Very good morning, children. Hope you have understood the last topic, that is cellular respiration. Okay, so today we'll deal one more topic which is called as anaerobic respiration. So we have seen that what is aerobic respiration and what is anaerobic respiration. So especially when you're talking about the aerobic respiration basically takes place in the presence of oxygen, and anaerobic respiration will take place in the absence of oxygen. Okay, so all of you concentrate here, okay, as we go ahead with the lesson. So when we are talking about the anaerobic respiration, we have seen that it happens in the absence of oxygen. Okay, it happens in the absence of oxygen. And second thing is that, so whatever the glucose is there, that is converted to, okay, that is converted to lactic acid, okay, converted to lactic acid, ethanol, or alcohol. So basically this is done by the bacteria and this is done by yeasts. And also we have seen that there are some muscles in our body which will convert the glucose into lactic acid when there is deficiency of oxygen. Okay, children, so this is the basic concept, right? So, and again in this, not only lactic acid and ethanol, apart from this, carbon dioxide is released, and at the same time heat is also released. So this is what we have seen yesterday, right? So what is anaerobic respiration? The respiration which takes place in the absence of oxygen and converts the glucose into lactic acid or ethanol is called as anaerobic respiration. Okay, so these examples are, it takes place in the bacteria and muscles, okay, and also in the yeast cells. Okay, yeasts are nothing but the fungi. They belong to the kingdom called as fungi. So this is the basic concept around what we are going to start the class now. So this concept you keep in your mind. So what is anaerobic respiration? Animals or muscles or bacteria or yeast, they take the glucose and they will convert the glucose to lactic acid or ethanol in the absence of oxygen. Okay, so that is what we call it as the anaerobic respiration, right? In this process, carbon dioxide is released and also at the same time heat is also released. So here we are going to do a small experiment by which we can prove that anaerobic respiration takes place and it gives out ethanol or lactic acid, okay, and also carbon dioxide. So based on these facts, we are going to do a small experiment. So this experiment is basically to prove that the yeasts, when they are put in the glucose solution, they will convert the glucose into ethanol or alcohol and also produce the carbon dioxide. So that is what we are going to prove experimentally. So theoretically we have learned till now, and now we are going to prove it experimentally. Okay, for this I have taken some apparatus here, so you can see on the board the apparatus which I have taken here. The first one is the thermos flask. Okay, for this experiment what I am taking here is the glass jar, okay, and also I have taken a test tube, and here you can see this black colored ones, okay, this is the rubber cork, Okay, the long one which I have taken is the thermometer, and the solution which is here is the lime water. Okay, this one is a bent tube. Okay, and the solution which I have taken here is the sugar solution. Okay, so this is the apparatus which I have taken to go ahead with this experiment. So first of all, what we will do is we will first prepare the glucose solution. The solution which I am seeing is the glucose solution. So first of all, we'll make out the glucose solution. Now this glucose solution will have oxygen in it, isn't it? This glucose solution which I have made here will have the oxygen in that, isn't it? So as it has the oxygen in it, so now our basic idea is that the respiration should happen here without the presence of oxygen, that is in the absence of oxygen. So this oxygen should be removed at any cost. So how it will be removed? A very simple procedure if you follow, you can remove the oxygen. So what you can take is, you can take this solution and you can boil it, okay, and then cool it. So boiling and cooling of this glucose solution will remove the oxygen which it has in it. So immediately you can put them in the glass beaker so that you can avoid it with the contact of the oxygen. So once you put it here, now you might have a doubt. Isn't it? So what kind of doubt you might have? You might have a doubt that this particular liquid will have the oxygen, isn't it? So this oxygen, whatever it is, this liquid which is over here, it might have the oxygen. To make sure that it doesn't have the oxygen, what you can do is, you can take the Dyson green. You can take the solution which is called as the Dyson green, okay, or you can call it as the Janus green, and what you can do is, you can put it in the sugar solution. So if the sugar solution turns pink, that means the level of oxygen is very, 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 very less. Only if the level of oxygen is very less, the Janus green which you pour in the sugar solution will turn into which color? Pink color. So by this we can confirm that the sugar solution which is here doesn't have the oxygen in it, or even if it has very, very, very low amount of oxygen it has. And then what we'll do is, we'll put some baker's yeast. Okay, you can put some yeast here. Now this yeast is a wild yeast which is usually grown on the fruits, here. So what happens is, so to eat this fruit, as it directly cannot eat the fruit, they will convert this particular fruit outside into liquid form, and then this yeast will absorb them. So they will have the fruit in this way. So those kind of yeast are basically grown on the skin of the fruits like grapes and apples, and from them they will get their food supplies. So those yeast we are collecting here and we are putting in the sugar solution. So, and moreover, what we can do is, also to avoid the contact of air, what you can do here is, you can take some liquid paraffin. At least 1 centimeter. You put the liquid paraffin. So this liquid paraffin, if it is above the sugar solution, it will not dissolve in that. It is a light, it's an oil light thing, and it will spread on the water, and it will not dissolve in the water. Okay, it floats on the water like this. So now this will avoid the contact of air with the water. So our water is now completely free from the air. So we have taken the liquid paraffin here, so that the air will not contact with the water, and we have taken the yeast solution here. Okay, and then what we'll do is, now this is the rubber cork we have put here, right? So this rubber cork, from this you will insert a thermometer here, so that it goes inside the water. It goes inside the water, right? That is in the sugar solution. And then you'll also arrange a bent tube in this way. So here, now we have kept this in experiment like this, and then the bent tube which you have bent here, you have taken, that will go and open into a test tube where you have taken some lime water. So this is the lime water which you have taken. Now this is the apparatus. Now our idea behind this is to make sure that in the aerobic respiration, the yeast convert the oxygen, the glucose into carbon dioxide, heat, and also alcohol. Now this is what we have learned theoretically. Now we are going to see whether this will happen practically or not. So if you keep this aside for some time, what you will observe is now the first thing what we can observe here is the yeast cells which are here, they are respiring. At the same time, when they respire, they also produce the heat. So the thermometer, the mercury in the thermometer will rise high, isn't it? If the thermometer, the mercury was here, it will go here. That means this particular respiration process, what it is doing here, it is releasing the heat. And second thing here is, now the gas which is coming out here, that will go through this paraffin tube and enters into this test tube, and converts this lime water into milky. What do you understand by this? If it is turning out into milky, that is now this bicarbonate solution, if this calcium hydroxide solution, which is nothing but the lime water, if this is converting into milky white, that means who is acting on that? Carbon dioxide is acting on that. So what's acting here? Carbon dioxide is acting, because only carbon dioxide, addition of the carbon dioxide to the lime water turns milky, and formation of the white precipitate. So now this is also clear, that is carbon dioxide is also released. And now you slowly remove this thermometer and also the bent tube and remove the liquid paraffin wax from the upper layer, and then you smell this one. When you smell it, the smell which comes out is exactly a alcoholic smell. So when you observe this one, so now the solution which is found here in the sugar solution, if you observe that, if you breathe it, it will give out a alcoholic smell. So as when it gives the alcoholic smell, you can say that this one here is converted to alcohol. So that means the yeast are able to respire even though there is absence of oxygen, when there is a very, very, very short supply of oxygen. And the same thing will be done by our muscles also. When the complete oxygen is depth over there, or if it is short in supply, then our muscles will convert the glucose into lactic acid and gives us energy. Here also you can see the yeast are converting into alcohol and also giving out the carbon dioxide. At the same time, they are also giving the heat out. So that is how we come to the end of this experiment. So now if you want to make sure that this is alcohol or not, alcohol boiling point is 70, whereas the water boiling point is around 100. So what you can do is you can go ahead with the fractional distillation method. This is the same method which is used to extract the different kinds of oils from the crude oil. You can see the picture on my right side. So what you can see is this is the place, refinery, where the crude oil is converted to different oils like kerosene, diesel, petrol, all these things, like LPG gas. So all these things, they come because each and every oil will have their own boiling point. So they will get into water vapor and they cool down at a certain point, and they will be filtered and they will be separated. So in the same way, you can also filter this ethanol from the sugar solution. That means the complete sugar will not be converted into alcohol, but only a little part of that will be converted into alcohol. And what you can do is you can go for the fractional distillation, and then you can separate this one and you can utilize it as a alcohol, right? So the same procedure is used in the beverage industries to make the alcohol, to make the beer, to make the other drinks which are full of alcohol. So not only that, you can also go ahead with the fermentation process, especially when you are doing dosa in your house, especially you are doing idli in your house. No, like night you'll put it as it is, and next day morning you can see the gas bubbles over that. And also if you keep it aside for 2 days, then you get a different smell, isn't it? You can get a sour smell, and that's the process which is done here is the fermentation process. Okay, so here the yeast, here the yeast they are able to respire, and they are able to give out the carbon dioxide, heat, and also the alcohol. That is how anaerobic respiration takes place. So anaerobic respiration also takes place in the yeast and also in the muscles. That is what we will confirm. Okay, by this we come to the end of today's session, children. So tomorrow we are going to see a very, very important topic, that evolution in gases exchanging system. So we are going to see how different animals have the different respiratory systems and all. So that is what we are going to discuss tomorrow. So be alert, and we'll meet on the next session.